Hello, and welcome to this webinar titled Identification of Approaches to Simulated Leachable Studies. What are they and when to do them? Hosted by Biopharma Asia magazine and presented by Jason Creasy, Managing Director at Maven E&L Limited. This presentation will be followed by an industry perspective presented by Karen Peters, Team Leader of Extractables and Leachables at Nelson Labs Europe. My name is Stephen Edwards, and I'll be your moderator. Before we begin, I would just like to inform our viewers that there will be a live question and answer session after both presentations near the end of the webinar. Audience members can send their questions in at any time during the webinar via the Questions tab located directly below your webinar screen, and we will go through these at the end of the webinar. Now, please allow me to introduce our first presenter. Jason Creasy is a graduate analytical chemist. He's recently set up as an independent consultant providing advice in the area of extractables and leachables after working for GSK in the area of extractables and leachables since the mid-1990s. Over that time, he has seen demand in his area grow exponentially, and Jason has held roles in increasing seniority relating to the support that GSK has given to extractables and leachables. Before setting up Maven E&L Limited, he was the director of a team of analytical chemists who were responsible for GSK's global R&D support for E&L activities across a wide range of product types and modalities. This included biopharmaceutical and small molecules, including inhalation, parentals, and cell and gene therapy use. He has had the pleasure of commenting on PQRI guidelines on E&L for GSK, the E&L section in EMEA guidelines on inhalation and nasal products, and co-authoring a chapter within a book entitled Leachables and Extractables Handbook, Safety Evaluation, Qualification, and Best Practices Applied to Inhalation Drug Products. Jason has been a member of several external groups concerned with the development of best practice guides for extractables and leachables. Issues these include the IPAC-RS Material Working Group, the BPOG, and continues as a scientific advisor to Extractables and Leachable Safety Information Exchange, otherwise known as ELSI. Currently, he is working and commenting on risk-based approaches to E&L requirements, which he hopes will form part of an ICH guidance in the not-too-distant future. I will now be handing over to our first presenter. Welcome, Dr. Creasy.
Thank you very much, Stephen. Thank you for that kind introduction. Today, I'd like to present approaches to simulated leachable studies, what they are, and when to do them. So the first part of my first slide here, just going over the content for today, I'd like to present on how they're different from extractable and leachable studies, what type of dose forms they might be best suited for, and I'm going to cover an example of a simulated leachable application. So, how are they different from extractable or leachable studies? Well, actually, they are just a form of an extractable study which aims to closely predict type and amount of substances found in the drug product. So really, they're a subset of extractable studies. They're not leachable studies, as they do not represent a measurement made in the drug product itself, but they are designed as a surrogate for those leachable studies. And in situations where leachable studies cannot be done, they provide an alternative. So as you can see from the Venn diagram here, trying to illustrate that the leachable studies and the simulated studies are very, very similar in terms of their output, and perhaps slightly different from the conventional extractable studies. So I've talked a little bit about that simulated studies being a substitute for leachable studies, but what truly are leachable studies? Well, leachable studies are generally recognized as samples taken directly from a drug product formulation. So in presenting them that way, they aren't necessarily the same as what a patient receives, and because the dose form can be a powerful influence. For example, with a topical dermal product, there's an example there that what leaches into the formulation is not very much similar to what actually reaches the patient, since the skin can be and probably is a significant moderator of that exposure. So there is a strong link here to bioavailability. That is the portion of the drug that enters the circulation when introduced into the body and able to take active event. So when we're talking about something that's administered intravenously, that would be a bioavailability very close to 100%. So leachable studies are themselves a simulation of what patient exposure might be. Moving on to extractable studies. Extractable studies are designed experiments, and how good that experiment is really down to the individuals designing those experiments. So what you might see in my illustration here is a number of different experimenters, and hopefully the Dr. Frankenstein figure is not so good at doing his experiments compared to a modern scientist working in a laboratory. So there's a number of assumptions that we need to build into a designed study, and a simulated study and an extractable study are just the same in that regard. There are assumptions made around the selection of the material. There is an effort to look at material preparation and make sure that it is an appropriate method for preparing the sample. That there are influences here around surface area, and will your experiment be a good model for what you're trying to simulate. Another aspect of this is the extraction solvent. The extraction solvent is a choice that you make, and in a simulation study, you're obviously trying to get close to the drug product formulation, and how close you can get to the drug product formulation will be very much dependent on the dose form and limiting factors around that. And finally, what I'd like to cover in this particular area is the temperature of extraction. You make a choice to run at a temperature that may or may not be different from the conditions that the drug product is used. If you do choose a temperature that's different from the conventional temperatures that the drug product is used at, you have to do that mindfully and be aware of the potentially profound difference that might take. So moving on, why not do leachable studies? After all, they are a very useful study to do. They are a direct measure of what is in the to be marketed drug product, or in the manufacturing system, if that's what you're discussing, or the drug product, if that's what you're looking to study. And as I've already explained, they are themselves a surrogate for what the patient is actually exposed to. Well, there's a number of reasons why it might be hard to do leachable studies. I'd like to go through some of those today. First of all, generally we're talking about a skilled process of analytical method development. You could have a situation where the drug product formulation interferes, or you could be trying to get to, most typically, very low detection limits in the case of leachable studies, parts per million or parts per billion even. And so to do that requires a considerable amount of skill when you're thinking about leachable studies. Every drug product and various dose forms might require a different skill set to be successful in producing those leachable studies. The second and also important aspect of this is, do you have the correct samples available? Very often, if it's a novel drug product and it's relatively early in the development of that drug product, the drug product may be in very short supply. It may be the focus there is to get into the clinic so patients can start to be exposed to the product, and sparing samples for a leachable study could be quite difficult. So that's one reason why you may struggle with sample availability. Secondly, you may want to study the variation from that you might want to see within leachable amounts, and in order to do that, particularly at an early stage, there may not be much variation in the samples that you have available to you. So a simulation study could potentially solve some of that by having access to more variation in the inputs. So coming back again to sample availability, clearly in order to conduct any sort of analysis, samples are very important, and as I've already discussed, availability of samples may be a difficult aspect of this, but it's not the only aspect. There's also time and the complexity of the formulation itself, and all of these added together really influence the skill set required and make overall leachable experiments quite challenging. So a simulation study may go some way towards reducing that complexity. Moving on, I'd like to talk a little bit more about what dose forms might they be best suited to. In principle, a simulation study could be applied to any dose form. It's just the nature of the simulation that may change. But in particular, they are applicable to one or two types of dose forms in particular. The first category is the complex formulation. And when I talk about complex formulation, I'm really talking about the formulations where the excipients and or the API itself make it quite difficult to actually measure leachables. Leachables measurements can be affected by many things, but having a formulation that is not amenable to GCS analysis or LCMS analysis, which are the two main analytical techniques that are used, means that you're going to have to look for some alternatives, and taking a simulation of your formulation and removing some of the more difficult elements of the formulation while still maintaining a simulation of the formulation is a possibility, and certainly is something to be considered, because if you can't measure leachables, then regardless of whether you're doing leachable studies or a simulation, you will have no data to present. So the next aspect that I'd like to highlight is one where the formulation might contain a biopharmaceutical, so large molecule, or human cells of some kind, such as occurs in biopharmaceuticals or cell and gene therapy. Both of these scenarios add complexity, which I've already mentioned, but they also add some some elements to this that make the sample preparation either very difficult or impossible. So in the case of biopharmaceuticals, we have very often a monoclonal antibody, which is very easy to denature as we take it through the sample preparation process. Any addition of heat or solvent will denature the protein, and once the protein is denatured, it may have a detrimental effect on your ability to measure accurate quantities of leachables present. So that is one thing that you might want to avoid when you're designing an alternative. The second category, and I'm going to speak about this in a little bit more detail in a minute, is cell and gene therapy. And in that application, you're actually using patient or donor cells to produce the drug product. And that makes a huge difference in terms of being able to conduct leachable studies, because those leachable studies will obviously be much more complex in the presence of cells and their associated media. So a simulation study is almost a prerequisite for doing that type of leachable study. So I just want to take you through how you might conduct a simulated leachable study in that context. So what I'd like to present here is a series of steps that I believe are necessary and useful in conducting a simulated leachable study for cell and gene therapy application. So the first step is a mapping of the process. And why that's important is because very often the process of production of cell and gene therapy product is a multi-stepped process, and being multi-stepped, it will involve the drug vehicle being in contact with multiple materials. Those multiple materials in turn can be a source of many and varied leachables. So the potential for leachables is quite high, but if you take the time to map the individual unit operations, you can obtain a list of the materials that are present, and you can come to a judgment as to whether those materials are going to be of concern in the process that you're going to risk assess. And when you consider risk assessment, you're considering the conditions that those materials are exposed to, and you're considering things such as time, temperature, and the composition of the fluid in contact with those materials. And in order to establish the potential for leachables, the consideration for the effect of time, temperature, and fluid composition enables you to write a potential, what I'm terming here, failure mode, and conduct a risk analysis based on failure mode effect analysis, where you're considering the probability and the hazard associated with that interaction. So you can actually score that risk in relative terms and determine which of the steps and which of the materials are most likely to be of concern as you're contemplating your simulated leachable study. So, as I've already mentioned, the cell and gene therapy area, which is showing rapid advancement at this point, is now using a lot of different processes to take the cells through their formulation and to introduce the type of agents that are then used to have a therapeutic effect on the cells. And in doing so, you're introducing many materials of different kinds. Some of them are plastic, some of them are elastomers. There are extended time periods, a range of temperatures. There's a whole host of different processes that may or may not be significant in terms of leaching substances from those materials. So without a systematic approach, it becomes quite challenging then to determine where to focus your efforts. But if you do take the time to break that process down, and here's an example of a typical process, you then have the possibility to understand where you might want to put your efforts, as it were, for biggest benefit. So what I've indicated here is that you have a couple of points here in the process that are higher risk for leachables and represent sampling points for samples that you can then collect if you were doing a sort of conventional leachable study. But what it also indicates here is that you have at this point to consider what the simulation study may represent in terms of conditions, in terms of time, temperature, and also be confident that the other unit operations here are of lesser risk, or they represent inputs into the process that is highlighted here. And therefore, in simulating this particular part, you have the opportunity to cover the risks that are present from the other unit operations at the same time. So the risk assessment process has to consider the fluid composition, what sort of solvent system is going to replicate the solvent system that the cell and gene therapy or any other process, any other dose form, if you're not talking about cell and gene therapy, might be. And then you have to consider, given that you're trying to adopt an analytical determination of leachables, that's your primary outcome here, is the solvent system going to allow you to maximize sensitivity and accuracy of that system? So that's another aspect of this that's really important in designing this study. Some solvents are easier to concentrate because they're volatile, the volatile nature of them, or their relative polarity allows them to be taken through some kind of pre-concentration step, and that can be very, very important in leachable studies to reach the required concentration. Purity is another aspect of this that shouldn't be overlooked. Some of the solvent systems that you might consider are very hard to obtain at a level of purity that enables you to be confident that your analytical procedure is going to spot trace level components within it. So consideration there and an appropriate choice of blanks and controls is really important. And then the formulation itself, which could very well be complex. Are there elements in that formulation that influence the rate of leaching of materials, such as surfactants or organic components, such as DMSO, which is quite common in cell and gene therapy application? And how are you going to then simulate or replicate that effect in the studies that you do? And lastly, how long and at what temperature are you going to have your material in contact with the fluid that you've selected? So the design of the experiment can actually come from the risk assessment that you have done. In fact, it should, in my mind, come from the risk assessment that you've done. So to a knowledge and an understanding of the process derives the best simulation possible. So one of the really significant aspects of this may well be the sensitivity that you want to achieve. You're probably going to try and determine how much exposure there's going to be, and what the analytes are, in order to conduct some kind of toxicological risk assessment. And with cell and gene therapy, you could have a very dilute solution in principle, and a large dose, a large volume being dosed. So that could get you to quite low concentrations. However, one thing to recognize is that this type of dose form is most often a small number of dosage units over a patient's lifetime, maybe only one or two, or a very small number. And therefore you have the opportunity to justify a higher AET, analytical evaluation threshold, which could well be very important in making the concentration a viable process. And the last thing I'd like to sort of make sure I cover here is the fact that validation of the methods is really important if you're going to rely on the numbers and feed them into a safety risk assessment. So my final slide really just to summarize some key points. Simulated leachable studies are a subset of extractable studies. They aim to be as close as possible to leachable studies. They're used when leachable studies cannot be done, and there's a variety of reasons for that. And I've illustrated that cell and gene therapy is one example where the conditions are really met for leachable, simulated leachable studies, because they're very hard, or if not impossible, to do in the conventional way. And I've also presented here today a technical risk assessment approach which can be very easily linked with successful simulated leachable studies, and just illustrating that simulated leachable studies also facilitate lower limits of detection and optimization of analytical conditions. So with that, I'd like to bring my presentation to a close, and thank you all for listening today.
Thank you, Dr. Creasy, for that great presentation. Now, before I introduce our next presenter, Dr. Peters, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. So please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the presentation. Now please allow me to introduce our second speaker, Dr. Peters. Karen Peters graduated as a bioengineer in chemistry in 2006, and after graduation she worked for 3 years as a quality document expert at Merck, Sharp & Dumb MSD, gaining experience in the regulatory and analytical challenges of pharmaceutical industry. In 2010, Karen started working at Topical Europe, now known as Nelson Labs Europe, as a study director for extractables and leachable studies, with a special focus on single-use materials used in the manufacturing of biological drug products. She is now responsible for a team of study directors specialized in extractables and leachable studies on large volume parentals, ophthalmics, and disposable systems used in manufacturing. She's presented on the setup and challenges for these applications at several international conferences. I'll now be handing over to our second presenter. Welcome, Dr. Peters.
Thank you very much, Stephen, for the introduction. Today, I will indeed also be talking about the identification of approaches to simulated leachable studies. So first, I will be going through some theory again, looking into what they are, how we can set them up, and then we will look into some case studies that I think might be of interest for the audience. So in the ideal case, Leachables are a subset of extractables. Jason Creasy already introduced us into the terms of extractables and leachables. And in theory, the components that you detect in a leachable study can be traced back to the extractable study. However, in reality, you will see that there often is a gap between leachable studies and extractable studies, and that in some cases unexpected leachables can show up in the leachable study. And this gap can be closed by performing a simulation study that includes parameters that are closer to the reality than the extractable study. So in the ideal world, an extractables and leachables assessment would consist of 3 steps, as was outlined also in the chemical assessment triad that has been proposed by Dr. Dennis Jenke, who is very well known in the field of extractables and leachables. So the first step is material screening and selection. So in this step, you want to select the materials that are the most appropriate for your application, and that can be by obtaining extractables data from your supplier or performing extractable studies under worst case conditions on the different components of your packaging system or your manufacturing system, and compare them between different suppliers, for instance. In the second step, a simulation study is performed where the parameters are closer to reality, but still worst case, to narrow down the list of extractables to a list of probable leachables. And then finally, product assessment can be performed where the presence of the leachables in the actual drug product under the real use conditions is examined. So this brings us to simulation studies. What are they actually? So a simulation study is closely related to a leachable study, but often the drug product is replaced with a simulating solvent. So typically the aging conditions are accelerated versus the real use, and the test article of a simulation study can be the complete packaging system or the complete manufacturing system as you would test it in the leachable study, or you can also focus on only a part of the packaging system. The concept of simulation studies was also introduced in USP 1663, that is the USP guideline on extractable studies. The purpose of a simulation study is then to detect and identify extractables that are probable leachables and generate an extractables profile that is more close to reality. So this will reduce the amount of compounds that are detected to those that are the most relevant for your specific application. And that will enable you to narrow down the list of potential leachables that you need to follow up in the final leachable study. So how can we set up such a simulation study? So like I said, often a simulation solvent is used, and the selection of that simulation solvent is a very critical step in the setup. Preferably, the composition of a simulation study simulation solvent should be as close as possible to the composition of the drug product. And parameters that you can take into account are, for instance, organic content, by summing up the percentage of organic compounds present in your drug product, but also pH and polarity. And it is very important that the choice of the simulation solvent is very well argumented and documented towards the authorities. Another pitfall of selecting a simulation solvent is that secondary leachables are not covered. So secondary leachables, these are reaction products between leachables and the drug product. Of course, if you're not using the drug product, these cannot be present in your contact solution. So if it is analytically feasible, it is advised to use the drug product itself, and this can be the case when your drug product is very simple in composition. So then we look at how we can select the appropriate conditions of a simulation study. So the conditions are preferably exaggerated and accelerated in order to be worst case versus the reality. The exaggerated conditions can be created by selecting a simulation solvent that is, for instance, slightly worst case in organic content or pH versus the real drug product. Or an increased surface area versus filling volume can be used. For instance, when you are testing bags, what you can do is you can fill the bag with a lower filling volume, so the surface area versus the filling volume will be higher. And this will enable you to reach, for instance, lower detection levels in the contact solution when you calculate the results back to the original filling volume. However, using such an approach with increased surface area versus filling volume also has some pitfalls, because if you exaggerate this too much, and the surface area versus the contact volume gets too high, the solubility of the extractables in the contact solution might get compromised. And then this might limit the concentration of the migrated compounds in the simulation solvent, because their maximum solubility was reached. And if you then calculate back the results to the original filling volume, the results will of course not be worst case anymore. Then in regards of conditions, so preferably accelerated conditions are used in temperature and time. So by augmenting the contact temperature, the contact time can usually be shortened. So after the conditions of the extraction or the contact simulation are set, the simulated extract can be subjected to an analytical package. And it's important that the analytical package contains methods for screening that are able to identify and detect a wide variety of different compounds. So here at Nelson Labs, we have a standard analytical package that contains headspace GCMS screening to detect volatile organic compounds, GCMS screening to detect semi-volatile organic compounds. Then we use UPL-CMS screening to detect non-volatile organic compounds. And then these screening techniques for organic compounds are complemented by ICP methods for elements. So we look standard for 32 different elements, including elements that are listed in ICH-Q3D guideline. And then we usually also perform an ion chromatography to detect anions. So besides this basic package of analysis, of course, other analytical techniques might be added based on a specific material. For instance, at the moment, there is a lot of regulatory concern in regard of nitrosamines. Nitrosamines cannot be picked up at very low levels with the standard screening techniques. So to detect these type of compounds, an additional analytical technique might be added. Another example is, for instance, silicone oil that is often used in pre-filled syringes. So for this component, we also have a specialized analytical technique, GFAS. So for some applications, as Jason already mentioned, the simulation study might partially or completely replace the formal Leachable study. This is, for instance, for drug matrices which cause a lot of analytical interference, and therefore screening at the required level is not possible anymore. For other products, such as the large volume parenterals, for instance, analytical evaluation limits can be extremely low due to the high maximum daily dose that is administered to the patient. And in this case, also the exaggerated conditions of the simulation study and the composition of the simulation study that is analytically usually more compatible with the analytical techniques can enable to reach lower analytical limits. For some packaging systems or manufacturing systems, they can be in contact with a very wide array of different products or solutions, and then a simulation study can facilitate to use a bracketing approach. Of course, when you are considering to replace your Leachable study with a simulation study, then it's very important to consider the regulatory acceptance. So can a simulation study be considered as a full alternative for a formal Leachable study? So when you are using a simulant like a mixture of ethanol with water, for instance, then it might be risky. So you will definitely need to well document the evidence that the simulation study, as you designed it, can well predict the results of the Leachable study. In addition, as I mentioned previously, formation of secondary leachables will not be covered by this approach. So this approach will only be accepted if the analysis in the drug product itself is really not feasible due to interfering matrices or extreme low analytical limits, and you will need to document this very well by, for instance, performing a feasibility study that justify the absence of the Leachable data. If you are able to use the drug product itself in a simulation study, then it might already get more likely to be accepted. However, typically you will use accelerated conditions, so those might not fully cover the migration behavior over the entire shelf life. So again, in conclusion, if you want to use a simulation study to replace a formal Leachable study, you will need to prove towards the authorities that you really have difficulties in performing a real Leachable study. So the implementation of the three-step approach, as it is mentioned in the chemical assessment triad, might have different interpretation for different applications. So the first step of the material screening and selection for a packaging system, for instance, can include controlled extraction studies on the components of the packaging system, while for a manufacturing system, this might be represented by a risk assessment on the different manufacturing systems that you use in the production process, and review supplier data on those materials. Then also the Leachable study can have different interpretations. So Leachable study can be a full screening study that is able to identify and detect unexpected leachables for a simple drug product, complemented by full quantification of target leachables with optimized methods. While for a complex drug product, it can be only the target compounds that can be followed up. So let's look at some case studies then. The first case study concerns a manufacturing line that's used to produce 25% human albumin solution. This solution has quite a high maximum daily dose of 400 milliliters, and that results in a fairly low analytical evaluation threshold that needs to be reached. When we start with the threshold of toxicological concern of 1.5 micrograms per day, we end up with an analytical evaluation threshold of 3.8 micrograms per liter. So because we are using screening analysis, we add an extra safety factor of 50% to take into account the analytical uncertainty of the method. So then we end up with a final AAT of 1.9 micrograms per liter. And because the solution contains quite a high protein content that causes high analytical interference, it will not be analytically feasible to reach that final AAT level by screening analysis. So for this example, we can follow a three-step approach, starting with the risk assessment of the different manufacturing systems that are used in the manufacturing process, and evaluation of the supplier data on these systems, then a simulation study, and ending with the targeted leachable study. So let's start with the first step, the risk assessment. So typically, bioproduction process contains a lot of different single-use systems that each may be made from different components coming from different vendors. So if you would need to subject all these components, all these materials to extractables and leachable studies, this will be a very challenging task, and it will be consuming a lot of time and resources. So therefore, typically a risk assessment is performed to select certain single-use system components for further testing. So instead of testing every single-use system for extractables, a risk-based approach can be applied that enables you to focus on those materials which might have a high impact on the leachables profile of the final drug product. So the goal is to select those single-use systems that have the greatest potential to introduce leachables into the final drug product that might have an impact on the safety, quality of your final drug product or on the process performance. And ideally, this risk assessment is performed as early as possible in the process development in order for you to still be able to introduce material changes where necessary. So this risk assessment starts with making a list of all the product contact materials. That means all the materials that make contact with the liquid process stream. And the list should be complete, starting upstream until the final finish line, and it may contain a very wide array of different materials. So once this list is complete, each material will be challenged versus different risk factors. This slide lists some of these potential risk factors. So this can be material compatibility. Does the material contain a lot of additives? Is it compliant with compendial chapters? And so on. Then the proximity to the final drug product or distance along the process stream is an important factor. Materials that are very much upstream have less probability to introduce leachables that will maintain throughout the manufacturing process. So often there are still dilution steps or purification steps that might diminish or remove the leachables from the production process stream. Well, of course, material which are present in the final filling line, they come in direct contact with your final drug product and also directly end up in the final container. Then the composition of the contact solution dictates the extraction propensity. For instance, the higher the organic content of the contact solution, the higher its extraction propensity will be. Then surface area to volume ratio. The higher the surface area of the system versus the volume of contact solution, the more leachables it will introduce. Temperature and time, obviously higher contact temperature and longer contact times will lead to higher concentrations of leachables. Pre-treatment steps should be taken into account, such as gamma irradiation or flushing. And then also some materials might be more likely to impact the process performance. So based on these risk factors, a numerical value on the risk associated with the material can be calculated. So to give some examples, for instance, a bioreactor is situated very much upstream, usually has a high filling volume, and therefore a low surface area versus volume ratio, and typically the risk associated with such a material is quite low. On the other hand, a sterilizing grate filter, which is situated in the final filling line, has direct contact with the final drug product and also has a high surface area versus volume ratio. So typically this is a more high-risk material. But of course, these are just examples, so it depends case by case on each production process. So for the materials that are then identified in the risk assessment as a high-risk material, extractable data will need to be gathered. In some cases, the supplier might have extractable data available, but one should be very careful when you evaluate this data to see whether it's suitable for your intended application. So parameters that you will need to check is the composition of the extraction solvents in terms of organic content, pH and polarity. Extraction conditions of time and temperature, are those worst case versus your real use application? Are pre-treatment steps covered, such as sterilization? And are the analytical techniques that the supplier used sufficient? So are screening techniques that can detect a wide range of compounds, and did they use a combination of different analytical techniques? So one should also always be very careful and keep in mind that the extractables profile is very dependent on the setup of the study. So if you compare data from different suppliers and they set up their study in different ways, then it's very difficult to compare them. So the data of the extractables study can then be compared with the analytical evaluation threshold. So if all the results of the extractable study are below the AET for manufacturing systems specifically, you might not need to do further follow-up. This is really specific for manufacturing systems. If you're talking about the packaging system, you will always need to do a leachable study. And also materials which are in the final filling line are more critical and will in most cases need a leachable study. But for materials in the earlier manufacturing line, if the results of the extractable study are all below 80, you might not need to go further. If there are some components or compounds present above the 80, you will need to perform further evaluation with a simulation study or a leachable study. So if we go back to our example of the 25% human albumin solution, the sterilizing grade filter came out as a high-risk material. Since the drug product was too complex to screen at the AET level, we will need to use a simulation study. And for this simulation study, we chose a simulation solvent of 40% ethanol in ultra-pure water. The composition of this simulation solvent was based on a white paper which has been written by Dr. Dennis Jenke. You can find it on our website. So it contains some examples of simulating solvents for some common drug solutions. And then the simulation conditions were set worst case versus the real use. So real use was 2 hours at room temperature, and that was mimicked by circulating the simulation study for 3 hours at 30 degrees through the filter. And also the extraction ratio was exaggerated by using 1 liter per filter instead of 6 liter per filter as it is done in real use in order to facilitate reaching the low AT level. The results of the simulation study can also be evaluated versus the analytical evaluation threshold. If the result of the compound is above the analytical evaluation threshold, but the compound is not considered to be carcinogenic or mutagenic, it can be checked versus the qualification threshold. In order to check whether you need to integrate it in the targeted leachable study. If the result of the compound is above the analytical evaluation threshold and there is a toxicological risk for mutagenicity, for instance, then you will need to select that compound as a target compound for your targeted leachable study. If the compound is present below the analytical evaluation threshold, and you designed your simulation study well to be worst case, then it should also be below the AET level in your leachable study. So in that way, you can select the target compounds that are relevant for this study, and optimized methods can be developed to quantify those compounds at the required concentration in the complex drug matrix. And you can use more high-end analytical instruments that are able to detect low levels in complex matrices, such as GCMS triple quad or LCMS triple quad. So then a second and last case study is about a bag system that contains a nutrition solution. So it's a 3-chamber bag where each chamber contains a different solution. So there's an amino acid solution, glucose solution, and the lipid solution, respectively. And then just prior to administration to the patients, these 3 solutions are mixed in the bag to an emulsion. So this solution causes high analytical interference because of the presence of the lipids, and it also has a high maximum daily dose of up to 2 liters. So the analytical evaluation threshold is extremely low. So if we look at the chromatographic data of the emulsion, then you see in the GCMS chromatogram there are really a lot of peaks. The internal standard is already difficult to find within this forest of peaks. So if you use this chromatographic data, can be used to document towards the authorities that the screening at such a low analytical threshold level is not possible. And then to close then the gap between Extractables data and Leachables study, a simulation study can be performed. So for this case, we chose 3 different simulation studies. So for the lipid solution, a 64% ethanol solution was used. So this choice was again based on the white paper of Dr. Dennis Jenke. And then in addition, 2 water solutions at pH extremes were added. Then the simulated conditions for this study were calculated using the ASTM standard for accelerated aging of medical devices. And in this way, the 24-month storage in the refrigerator was mimicked by 4 weeks of storage at 55 degrees. So using both the ASTM standard and the white paper, the use of the simulation solvent and the simulation conditions can be well documented. So in conclusion, simulation studies can be used to close gaps between extractables and leachable studies, resulting in a 3-step approach, including extractables assessment, simulation study, and then a final leachable study. For some applications, the leachable study can be partially replaced by a simulation study. However, if you do so, you will need to provide proof that the formal leachable study is not possible. In addition, it's very important to well document the choice of your simulation solvent and the conditions towards the authorities. So this brings me to the end of my presentation. So I think we are ready for a Q&A session now.
Thank you, Dr. Peters. That is correct. Now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question will be for Jason. Our question is, do the methods used in simulated leachable studies need to be validated?
Hi, thanks for that question. The short answer to that is yes. I think the longer answer is there's a variety of different ways that you can validate methods, and it depends on what the data is going to be used for as to the type of validation and the quantity of validation that's going to be necessary. I mean, the one that I pointed to in my presentation was the need to perform a safety risk assessment. Now, in order to perform a safety risk assessment, you have to have an idea of what something is, and you also have to have an idea of how much patient exposure there has been. So the validation aspects of that speak to being able to say that your method is specific and saying that your method is accurate, and to have a degree of precision, which are all elements of validation. But the amount of precision that you might have for a safety assessment may not be the same requirement as you would have for a routine evaluation of against the specification, for example, because the safety assessment is really adds a lot of assumptions into it, and therefore the numbers don't have to be as precise. But it's really important that it is accurate in the sense that you're safety assessing the right material, the right substance. So it really does depend on what you're validating for. But generally speaking, yes, they need to have some element of validation associated with them.
Thank you. Moving on, our next question is for Karen. Next question is, if the drug product does not interfere too much and the final AET is not extremely low, why would I still perform a simulation study?
In some cases, the extractable data that are generated worst case and under general conditions. So in those cases, the data that you obtain might not be so relevant for your final application. So when you're designing an extractable study, really worst case, because you don't know the final application yet, for instance, you might end up with a really long list of compounds. And then if you use a simulation study, which has then parameters which are more closely related to your specific application, then that might enable you to focus on those compounds that are really relevant for your application to follow up in a leachable study. So if you have to use extractables data, worst case extractables data as a basis for your leachable study, you might end up invalidating compounds or a high number of compounds that might not be so relevant for your final application and might never be present in your real drug product. So in that way, the simulation can facilitate in that and can even save you money, because you can validate less compounds and the most relevant compounds for your application.
Thank you. Moving on, our next question is for Jason. Our next question is, do all regulators agree when simulated leachable studies are required?
I guess this is the way I think about this is that there's a sort of broader question around extractables and leachables, and do all regulators agree? And I think the short answer again is no, they don't. I think it's becoming better as more guidance comes to out there. I don't think it's going to become substantially different until we have an ICH guidance, which I'm hoping will happen over the next few years. I think there's reasons why regulators disagree. I think there's a lot of variation between different dose forms. It's hard to come up with sort of a one-size-fits-all approach here. I think you've got to be guided by best scientific approaches, and then the regulators will kind of fall in behind that. I think this is a sort of general point here with regard to simulation studies. It's the quality of the data that you obtain that really drives that question around when they're applicable and when they're not. I think the general answer is no, the regulators don't agree, but I'm hopeful for the future.
Thank you. Our next question is for Jason, for you again. Our next question is, I believe this is in reference to your presentation, are they better than actual leachable studies?
Simulation studies, are they better than actual leachable studies? Well, it depends is the answer to that one. As I've just said, I guess it comes down to what quality of data you're able to achieve for either a simulation study or a leachable study. I think by preference you would, and I think Karen's presentation said this as well, by preference you will try to achieve a leachable study, a measurement in the drug product formulation. But where it is not possible to do that, or the quality of the data is compromised by trying to do that, then simulation studies are better. And my study, my presentation indicated that there are certain situations, certain applications, and cell and gene therapy was my example, where it is not practical or possible to generate a meaningful leachable study. And in that instance, simulation studies are better because they generate quality of data that makes that evaluation of leachables possible, where otherwise it wouldn't be. So it really comes down to that one thing around what is the most quality, what's the highest quality you can get. If it's from leachable studies, do your leachable studies. If it's from simulation studies, do simulation studies.
Thank you. Our next question is for Karen. Our next question is, did you have cases where the Leachable study was replaced by a simulation study and the study got approved by the FDA?
Yes, we did have cases, but like I said, it's very important in that case that you well argument why the Leachable study was not analytically feasible. So you should preferably really have analytical data that supports that the Leachable study was not feasible, and that at least you tried or you attempted to analyze at least some targets in the actual drug product.
Thank you. Our next question is for Karen again. Why would one still perform an extractable study and not immediately start with a simulation study, as it is more close to reality and also provides us with information with regard to the potential leachables?
Yes, this is often indeed something that is being considered by customers if there's not enough budget or if there's not enough time to perform a full ideal three-step approach. So it is possible to skip the worst-case extractable study and then start with a simulation study immediately to prepare for your leachable study. If, of course, your packaging system is already fixed and you know already which materials you are using. However, when you immediately start with a simulation study, where you use a complete packaging system and fill it with a simulation study, simulation solvent, for instance, it will not be possible to trace back leachables to the individual components of the packaging system. For instance, if you have a bag system, for instance, there are a lot of different components in a packaging system. You have a film, you have tubes, you have connectors, printing inks, and overpouch. So they all might introduce leachables in your contact solution. So if in the leachable study then a compound of concern is detected, you will not know from which component it is originating, and you might still need to do extractable studies on the different components separately. So yes, in that regard, it is a bit a risk, but it is possible.
Thank you. Moving on, our next question is: do you perform for the extractable studies / leachable studies sampling only the end of the studies, or do you also perform sampling in between? And do you think it would be necessary? And if not, why? Karen, would you like to answer that question first?
Yes. Well, I assume with extractable studies, they are referring more to simulation studies. So typically in a simulation study, you use more accelerated conditions. There's usually only 1 or 2 time points under accelerated conditions. This, of course, is a bit of a risk because some compounds may need time to migrate. So if you're performing a true formal leachable study, then I would really advise to include multiple time points over the entire shelf life. And we also know that this is required by FDA. So the FDA published some guidance on that. I think it was a bit more than a year ago, so that they really expect for a formal leachable study that you include multiple time points over the entire shelf life, so not only the end of shelf life.
Thank you. And on the back of that, Jason, do you have anything you'd like to add?
I think the only thing I would add is that it really comes down to how much knowledge and understanding of the system that you have as to how you sample. As Karen points out, the extractable studies and the simulation studies, they really come from a position of understanding or simplicity, so they may only just have one or two sampling points. Leachable studies, you may not really have much of an insight into what the relationship is between time and temperature and storage conditions. So that's why you might be sampling over multiple points so you can plot a curve and see what the trend is. So it really comes down to knowledge and understanding as to how you sample.
Thank you. And moving on to our next question, I believe this one is more for you, Karen. You talked about below AET. How are the extractables quantified?
Yeah, so in an extractables or a simulation study, we typically use screening analysis. So these are methods that are designed, these are generic methods, and they're designed to detect a very wide range of compounds. So it's not a full quantitative method, it's what we call a semi-quantitative method. So an internal standard is spiked to the sample prior to the analysis at a known concentration. The compounds that are detected in the extract are then quantified by comparing their analytical response to the analytical response of the internal standard. So it's a semi-quantitative method because you are supposing that the analytical response of the compound that you detect is the same as the internal analytical standard, which will of course not be the case for all compounds detected. And that's why in the calculation of the AET for screening methods, we always build a safety factor to deal with this analytical uncertainty.
Thank you. And with that, thank you for your answers, and thank you, audience members, for your questions. Before I finish the webinar, I would like to ask our presenters, Dr. Creasy and Dr. Peters, if they have any closing remarks. So, Dr. Creasy, do you have any closing remarks for our audience today?
One thing I would say is it all comes from knowledge and understanding. If you have a good knowledge and understanding, you can do this. Without it, you're struggling, whatever you try and do. Simulation studies, leachable studies, extractable studies. So you have to take time to design these things properly.
Thank you. And Dr. Peters, do you have any closing remarks for our audience today?
Well, indeed, I agree with Dr. Jason Creasy. It's all about understanding what you are doing and also argumenting it in a good way towards the authorities. If the authorities can follow your reasoning and your rationales, they will, of course, more likely to accept it. Because we sometimes see that studies are being well designed and everything, but if you don't document it well and don't write down well towards the authorities what is your understanding behind it, then it is of course difficult for them to follow, and it will be more difficult for them to agree.
And with that, I would like to take this opportunity to thank our presenters for sharing their knowledge with us. Once again, everyone, that was Jason Creasy, Managing Director at Maven Extractables and Leachables Limited, and Karen Peters, Team Leader of Extractables and Leachables at Nelson Labs Europe. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send any extra questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone, and have a great day.